Okay, this one is also a comparison between the innate and adoptive immune system. Certain simple things we have already gone through. You can just browse these slides for you. There are different types of adoptive immunity we are having. Number one is called as humoral immunity, and the other one is called as the cell-mediated immunity. The humoral immunity is understandable, like a problem is there in the bloodstream. Usually it belongs to the problems in the bloodstream, because the bloodstream is like a vast area. If you can assume in your head that the bloodstream is like an ocean. I mean, everywhere in our body. So when a problem is there in the bloodstream, it's like everywhere. It's like moving from head to the limbs, and from the limbs to the legs, like moving everywhere. If the trouble lives in the blood, then we need to fight that thing by releasing certain responses within the blood. And that stuff and the job of that thing is done, usually carried out with the help of the beta cells. You come beta cells. So usually the extracellular microbe, they use the term here, the extracellular microbe. I think something is not present between the cells. Something is present in the lymphatic system. Something is present within the bloodstream that is away from the cells. So usually that we cannot deal with those things unless we send the barriers, we send the response into the bloodstream. And that can only be done, or most of the time it will be done with the help of the antibodies produced by the beta cells. That is, it's called humoral immunity. Other thing is one very specific, one-to-one, eyeball to eyeball. It's like killing the bacteria by looking at the bacteria. It's not like producing the antibody and sending the antibody to the bloodstream. Jahan pe trouble hogi, wahan pe wo usko maar dalega. Humoral immunity. But in case of cell-mediated immunity, it is like cells are trained, find the bad things, capture it, finish it. It is called the cell-mediated immunity. And that is done, it is involved in phagocyte. Phagocytes are the cells which can do phagocytosis. Phagocytosis is the ability of a cell to eat the microorganism. एक तो हम directly eat कर रहे हैं ना, इससे भी of course external bacteria will be dead. जब macrophage इसको खा जाएगा तो वो मर जाएगा. But macrophages are always less in number as compared to the microbe. वो bacteria thousands में produce होते हैं. Macrophages are limited. They cannot finish the bacteria by eating them. It's not enough. Even अगर एक macrophage 10 bacteria भी खाए, it might not be enough to kill the bacteria, because the rate of replication of bacteria is very faster as compared to the number of cells we are having in our body. So that's why it's not possible. But the one way is to eat the thing up. Macrophages or phagocytes are doing those things. The other is the activating the T cells and the beta cells. Once the bad cells, or I must say, once the macrophage contains that information, like this macrophage contains that information can be transferred to the T cells. The T cells will got agitated, and then they start doing their function. They will start killing the bad thing. There are other cells. The T cells can also produce another type of T cell. This is just another type of T cell called cytotoxic T cell. Just you don't need to remember the name at this time. It is cytotoxic T cells. T cells can produce the cytotoxic T cells as well. The cytotoxic T cells are capable that whenever they find the bad thing, they capture it and they kill it. It is called, I used to call it like, it's like a kiss of death. But they are very specific. They know where to go and what to kill. And it is used to kill the infected cells. This is very important. The cytotoxic T cells are designed to find what? To find the infected cell. A cell is harboring the virus. A cell is harboring the virus. That is an infected cell. A cytotoxic T cell is capable of capturing that cell. It can capture the cells and then it can kill that cell. It's very special, very important. We do not give that ability to the other cells. हम ये पसंद नहीं करते कि किसी सेल के पास ये ताकत होगी वो दूसरे को मार डाले. I mean, it's too much power to be given to somebody at a cellular level. I mean, हम हर एक को पिस्टल या हथियार नहीं देते हैं, पुलिस को देते हैं ना ट्रेनिंग वगैरह करने के बाद, because the police may know कि किसको मारना है किसको नहीं मारना. You don't give this thing to everyone. Similarly, the primary weapon, or I must say a very special weapon, was given to the T cell, which became like a cytotoxic T cell after a lot of training and education. किसको मारना है. But think, this is not humoral immunity. This is a cytotoxic T cell which is capable of killing an infected cell. It will not go to the bloodstream. It will go and find the infected cell. Infected cells कहाँ हो सकते हैं? Hepatitis C virus हुआ तो infected cells liver में होंगे. अगर लंग डैमेज हो रहा है किसी इन्फेक्शन से तो इन्फेक्टेड सेल जो है वो लंग्स में होंगे, दे विल move with the bloodstream and then pass on to the site of the trouble, and then they kill and destroy only the cells which are harboring the bad information, which are infected. So that is how they're able to kill the bad thing. And it is completely different than the, that is like firing. Humoral immunity is like firing, like open fire to the enemy. Just whenever they find the bad thing, they will kill it in the bloodstream. In cell-mediated case, specific cells are created, they go and find the bad cells and then they kill them. Because we cannot capture the virus and kill them in a way we can kill the bacteria. Bacteria can be killed more easily, quite big, visible. But viruses, they are like very small. It is very difficult to identify the virus. So our body develop a way, instead of identifying the virus, how about identifying the infected cells? Virus को identify करना जरा मुश्किल है, तो how about identifying the infected cells? ताकि infected cells मार दें, they will not going to contribute in spreading the infection, and that is how we might be able to finish the virus. That is one of the way.